Right, so cones, color cells. So let's discuss now hearing. I think this is where we ended. So I didn't want us to get too much into hearing because by the time you guys were done on Monday, I could tell you guys were exhausted. All right, and with hearing and equilibrium. Hearing, balance, and equilibrium. Now, hearing has to do with certain subsets that we see in our body. First of all, the nerve is different, and when we look at this structure right here, remember, you actually have two different structures. The cochlea is on one side, and then the semicircular canal, along with the utricle and saccule. So the semicircular canal, that's for balance and equilibrium, and then the cochlea are specialized for hearing. So what we're going to see then is that each of these structures are going to go through different nerves. So I want you guys to understand that there is the vestibulocochlear nerve are actually two separate nerves. So the vestibular nerve is one nerve, the cochlear nerve is one nerve. They don't ever intermingle, right? They're always separated. It's not like the facial nerve when you have like the facial nerve and there are branches of it. Well, this nerve for balance and equilibrium here, hearing, well, those vestibulocochlear nerve are two separated nerves continuously. The cochlear nerve comes through the cochlea. The cochlea itself is snail-shaped. And what we're going to see is that it starts at this broad area called the vestibule. So let's kind of discuss this anatomy first. Now, I know we discussed it when we talked about the ear when we went through the model on Monday. Again, right? When we're looking at this, it's going to be on video. So, right? So we're going to hear that you have amount of gases on each side of the tympanic membrane. At sea level, we are at 1 atmosphere of pressure, 760 millimeters of mercury. Remember, when you guys, from now on, in chemistry, in physics, or other things you have to take, whenever you see the word pressure and see it as it relates to a gas, pressures in a gas is how we measure its concentration. So we can't see the gas, so we can't really see how many moles there are. So how can we tell how much of a gas we have? Well, we can tell by how much pressure it exerts. You have a lot of that gas, you're going to exert a lot more pressure. So what happens? At sea level, all of the gases in our atmosphere, nitrogen, all right, that's about 79%. Oxygen, about 20%. And then the remaining 1% is a combination of carbon dioxide, water vapors, right? Those gases are found in those concentrations throughout our atmosphere. But at sea level, we usually have the greatest concentration of them. At sea level, all those gases combine to give us a pressure of 760 millimeters of mercury. And what happens then is, as we are climbing, and you've heard of people that are climbing mountains and they get altitude sickness, right? They get really sick. Sometimes they even die on the mountain because of altitude sickness. What happens is, as they're climbing, the increase in altitude, there's less and less atmosphere. So the concentrations of the gases are still the same, there's just less of the gas. So instead of having 760 millimeters of mercury, as we climb up, you're at half of that. If you half the amount of oxygen in the atmosphere, well, you're gonna be in serious trouble, aren't you? You're decreasing by half just by climbing. By the time you get to 8,000 feet, which is like, near the very top of what we call the death zone in Everest, right? At 8,000 meters, what happens is the atmospheric pressure is so low that if you don't have oxygen or don't have just gas being pumped in through a tank, you will die if you're there for more than 10, 15 minutes. Right? And what happens is, because of the pressure drop, there's less available gases. And here at 1 atmosphere, at sea level, there's 1 atmosphere pressure in front of the tympanic membrane, 1 atmosphere behind the tympanic membrane. Right? What happens? We're at equilibrium. Tympanic membrane is free to move around. As the tympanic membrane vibrates, is gonna vibrate differently depending on whether the sound we're hearing is of high frequency or of low frequency. So if it's a high frequency sound, I'm just gonna move up here to show you the frequencies, right? If it's a high frequency sound, it causes your tympanic membrane to vibrate faster. If it's low frequency sound, like your bass sound, usually males have a lower voice. What happens is the tympanic membrane and the sound waves vibrates at a slower oscillation. Now, that allows us to hear the different frequencies. What happens is the way that the tympanic membrane, eardrum, starts to vibrate, whether it vibrates very slowly or very fast, we will then have that movement of the tympanic membrane being transmitted to the malleus, incus, and the stapes. In the stapes will then start to vibrate at the same oscillation. And what we're going to see in the stapes is that the stapes right here will make contact with this membrane called the oval window. This membrane found in the oval window is a membrane, like a very small version of the tympanic membrane. And what we're going to see that the oval window region right here, there's some fluid behind that area. So as the stapes starts to shake, the membrane found in the oval window is going to cause vibrations throughout the fluid deep to it. And what we're going to see is that certain regions of the cochlea have hair cells that are there for certain frequencies. So at the base area right here, the hair cells there, if they become active, will be very important for the higher pitches. The hair cells at the tip of the cochlea, if they activate and bend, they will then get activated and bent, and you will hear the lower end of our frequency spectrum here. So what we're going to see is, depending on where those hair cells are going to be found, they're going to give us the ability to hear the different pitches. If the hair cells are found at the base and they bend, you will hear the higher ends. If the hair cells are found at the tip, the helicotrema, those hair cells, if they bend, will allow us to hear the lower ends. And we're going to see that most of us lose the ability to hear the higher ends, right? It's a natural part of our development as we age, is that we lose ability to hear the higher ends. Hopefully we don't most of us don't lose the ability to hear the lower ends. Now, when we hear of people becoming deaf, losing hearing, the problem is we're not worried about the very, very high ends. We're going to lose those. It's the mids that we're worried about, right? The mids that allows us that most people are not going to be super high soprano voice or super low, right, very white voice. So what happens is we're usually in the mid-tone region. If you start losing those, you lose the ability to hear what most people are saying to you, which is the worst possible situation. So again, right, we're always talking about movement of the tympanic membrane. The faster the tympanic membrane moves, the faster the malleus, incus, and stapes start to vibrate and shake. As it starts to vibrate and shake really quickly, the stapes is connected to that oval window. It starts also shaking the oval window. The oval window shakes with that same frequency. And as it starts to shake, it causes fluid to start to move. And it's gonna then move at the same frequency. And eventually, we're gonna cause that fluid movement. It is that fluid movement that eventually causes our hair cells to bend. And we'll talk about how in just a second. So keep in mind, at this point, I'm just setting up that stage for understanding hearing here. And this model, in this chart right here, you see the function of the stapedius and the tensor tympani. Now the tensor tympani muscle, the name kind of explains what it does. It's connected to the malleus, which is connected to the tympanic membrane. Its main purpose, these two muscles, Main purpose is to stabilize the bones, stabilize the tympanic membrane, so then you don't burst the eardrum from loud sounds. Now, there are loud enough sounds that overwhelms the tensor tympani. They overwhelm as well as the pediis muscle here. And what happens is, if you have a very loud, sudden explosion, well, that's so sudden, it's so loud, that there's no way you can prepare for it. What happens then? Your tympanic membrane is really gonna move with a lot of amplitude. Because it's a loud sound. The louder the sound, the more amplitude, right? The more the tympanic membrane, the depth that it moves is going to be greater, right? So instead of just kind of vibrating kind of slowly or fast, but you know, if you're having a very whispering voice, it vibrates, and the actual tympanic membrane doesn't move that much. Sudden sounds, you're gonna move the tympanic membrane a lot, to the point where it can burst, to the point where even the bones can break. So you've heard of people going deaf because of a loud sudden explosion. Well, those people are gonna burst the tympanic membrane and damage the malleus, incus, and stapes. Sorry about this, Shanae. I have the camera right there, ball right in front of you. Right, so now let's talk about this oval window right here. And what we see in the model, the board model, over here, right? Now what we see of the board model is this. How can we see the cochlea here? And how do we translate that cochlea, something that appears to be circular and continuous right here? How come we see this then? Right? What we see of this is simple, all right? This is a cross section of one part right here. Now, I want you to imagine that this is just one cross section. That if you see it in the cochlea, this kind of spirals all the way up and around, right? As the cochlea is a combination of all these cross sections spiraling up and around. At the very tip, you have the helicotrema. So this is one section that we're looking at of the cochlea. When we cut it and look at it, there are three parts, right? Right up here, three parts. Now here on this slide, they just discuss what's found there, the fluid that's found there. In a second, we'll talk about what the name of those compartments are. So when we take a look at this, I'm just gonna try to pull it up so we can see it here. And then I'll show it to you and explain it to you on the picture first, and then on the model, right? On top, so you have three distinct compartments. It looks at this point like the top compartment and the bottom compartment are completely different. No way does it look like it's connected, right? Well, in a second, I'll show you what it looks like when we unroll the cochlea. Because remember, it kind of spirals up, right? What happens when we unroll the cochlea? What does it look like? In a second, when we unroll it, we're gonna see that the top compartment and the bottom compartment is actually one compartment that bends around. But because of a cross section, it looks like it's two separate compartments. So let's take a look. The top compartment is called the scala vestibuli. That is number 2 in our model here, right? Then you have the bottom compartment called the scalae tympani. That's number 3 in our board model. The middle part is the area that actually contains the cells that allows you to hear. That middle part is called the cochlear duct, sometimes also called the scalae media. So that middle part that's pink right there, that is the compartment that's most important. Whoops. Right? So hopefully everybody wake up from that. Sorry.
Yay.
Gonna edit it out. Right? So the top part right there and the bottom part are one continuous unit that kind of folds around. The middle part contains the cells that allows us to hear. So let's talk about how we activate this middle part then. The middle part region right here contains hair cells. And we've talked about hair cells when we talk about taste, when we talk about smell. These hair cells, right here, they have two different parts, two different types. You have the inner hair cells, because they're on the inside. It is number 8 on the models. And then you have the outer hair cells. The inner hair cells are the one that's important for hearing. So when we look at this, the inner hair cells are number 8, the outer hair cells are number 9. The inner hair cells, if you take a look, both hair cells at the bottom region right here, the bottom region, they are going to be anchored in place to a membrane called the basilar membrane.
Basement.
The basilar membrane is going to be number 6 on our models list, right here. So that's the basilar membrane right there. And what's going to happen is this. Since the inner hair cell and the outer hair cell is sitting on the basilar membrane, and since it's got little projections, little hairs we call stereocilia, those little hairs are touching and embedded on a different membrane. That membrane is called the tectorial membrane right there. Right? The reason why we're going to talk about this is this. And it's really important to understand. In order to activate the hair cells, we need to physically bend those hairs. We need to physically cause a shearing and a bending of those hairs, or the stereocilia of the inner hair cells. So how do we physically cause it to bend? So what we're going to see is this. As we start talking and hearing, we're going to cause fluid movement. The fluid then starts to move through the scala vestibuli. It's going to start to move and vibrate into the scala tympani. As the scala tympani starts to vibrate, the fluid there starts to vibrate, it's going to cause the basilar membrane to start to move. As the basilar membrane starts to move and shake, well, the inner and the outer hair cells are connected to the basilar membrane. They're sitting on the basilar membrane. So as the basilar membrane starts to shake, the inner hair cells also starts to move. The bottom of the hair cell starts to move. Right? As the hair cells bottom start to move, the tip is embedded on the tectorial membrane. What happens if you move the bottom, but not as much the top? Well, you start bending, right? This is exactly what happens here. As we start shaking the basilar membrane starts to move, causing the inner hair cells to also move at the very bottom, where it's connected to the basilar membrane. So the base of the basilar membrane right here, and the base of our hair cells, they start to move at the base, at the bottom. But the tip of the hair cells, the tip is embedded on the tectorial membrane. That doesn't move as much. And what happens? As the bottom moves and the top doesn't move as much, what do you cause? Bending. Right? And as we physically bend the cells, we cause those cells to bend. And depending on where those cells are that bends, right? If the cells that are bending are found at the base, all of a sudden then we have the high frequency sound, right? If it's the hair cells are found at the tip, then we can hear the lower frequency. And an action potential is fired into the cochlear nerve at that point. Now I'll show you a video in a second, and the video is good because it shows animations, and if I could draw, it'd be a little better, but since I can't draw, I have to use other people's animations. Alright, so here's this cochlear duct in the middle. Again, cochlear duct, also called the scala media. Cochlear duct filled with endolimb, which has high potassium. Now, what is important about this? Only place in the body where potassium in causes an action potential. No other place in the body where potassium influx actually causes depolarization. Now, normally it's sodium, right? Influx. This is the only region that allows us to have potassium influx causing an action potential. So that makes it pretty rare. Hint, hint, hint. right? So when you hear something that is that factual, that specific, you gotta think that's gonna be in a test, right? So I want you guys from now on, when you're taking a test, when you're reading, all right, when you're something that specific, you need to focus in and, you know what, I'm gonna take an extra minute here, memorize this, just in case it's on the test. Now, we also mentioned for the third or fourth time about where you find the inner hair cells. If the inner hair cells are found near the base, well, if they activate, they allow us to hear the high frequencies near the oval window, near the base. Near the apex, the lower frequency vibrations. Now, in my other class, I described what happens as well in terms of the ability to not hear the higher end. Anybody has a dog at home? Anybody ever heard of a dog whistle?
Yeah.
Right? Anybody's ever heard a dog whistle? Right?
No.
Usually you don't hear the dog whistle. The reason why? Because the whistle is at such a high frequency that dogs, with their increased ability to hear, can hear it, but we can't. Right? So what happens is we don't activate the basilar membrane in those regions. Then the hair cells in the higher frequencies do not then activate. Now, what we're showing you here is why that bending of the hair is so important. Bending of the stereocilia caused by movement of the basilar membrane and the slight movement of the tectorial membrane. The combination allows us to bend the stereocilia, which opens up spring-loaded channels, and you're mechanically popping it open by bending. So by bending it, you open up the gate, allowing potassium influx. Again, potassium. As that happens, you depolarize the hair cells in that region, allowing you to hear that frequency. Now, if you damage the hair cells, you will start having a little bit of deafness. The good news now is this: as long as a cochlear nerve is not damaged, now it's become much more prevalent to see people with a cochlear implant device. A cochlear implant device, it's got this little round thing that goes into the actual nerve, and then it's got a kind of, follows the ear, right? Kind of like a hearing aid effect, right? And that allows you to decipher the sound waves coming at you. It's pretty cool. And now, in the old days, it used to be huge. Now it's gotten smaller, and now we know that because it's so effective, more and more kids that are deaf are able to use it. And again, when we talk about frequency, it's about how fast the sound wave oscillates. Also, how fast the tympanic membrane moves. Now, you guys have heard of people that have high-pitched voices, the soprano voices, and you can break glass, right? Well, that's really hard to do, but sometimes, not with our own voice, but with some kind of mechanical help with a computer voice, we can cause glass to break. Now what happens is, in order to do that, you need to have usually a high-pitched voice that's very, very powerful. High amplitude, high pitch. Why does it cause your eardrums to burst? Because what happens when you have high amplitude and high pitch is it causes your tympanic membrane to move back and forth with a lot of depth. Not just back and forth with a lot of depth, but fast back and forth with a lot of depth. The speed allows us to have that higher pitched sound. The depth at which the tympanic membrane now starts to move is the amplitude, is the loudness. Right? So if you have something that's loud and super high pitch, you're vibrating the tympanic membrane so much and with such amplitude that it can't burst. Remember when I mentioned that the top part is connected to the bottom part? Here's what the cochlea looks like when it's unrolled. So this area right here is near the vestibule. This is the area of the oval window. What happens? Right? Now, remember, when we talk about sound, you guys have heard about geeks and nerds talking about Star Wars. I say that, I'm a geek and a nerd, so it's okay, right? So what happens, they talk, and you always hear about, people complaining, like in what is not The Last Jedi, but The Force Awakens, right? The first Star Wars movie after like 10 years. They talk about what happens when there's a bomb, right? You drop a bomb in space and all of a sudden you're supposed to hear an explosion and people are like, why are you hearing an explosion, right? Obviously it's because it's a movie, right? Because they were complaining because if it's in space, there's no matter in space. It's a vacuum. There's no air, there's no gas in space. So in order to hear something, you need some kind of physical medium. You need air, right? Here, I'm moving air. The air then hits your tympanic membrane, causing it to move. But what happens if there is no air? If there is no air, there's nothing to move. You don't hear anything. Water is a physical medium. Water actually transmits sound better than even air. Now, it's hard to talk underwater because you might want to, breathe instead of talk, but there are certain things that divers do. So next time you're at a pool, go underwater and start just hitting your hand like this underwater. When you hear, it doesn't sound like anything. But even if you do it to just this small extent, you're gonna hear, and people 30, 40 feet, right, in front of you will be able to hear that sound. If you're outside and I do this, there's no way you can hear that now. But because water is such a great medium, when I do this underwater, people can hear it all over. And that's what divers use as a way to signal when they should come up, right? Because they've been there underwater too long. Now what happens is, why am I explaining it like this? Because we have fluid here in the middle ear, in the cochlea. And as we vibrate the tympanic membrane, the malleus, incus, and stapes vibrate the oval window, which causes a vibration to go through the fluid. And remember, as the vibration goes through that fluid is like water. It carries that sound waves even better than air does. So as that sound wave for the high pitch starts to vibrate and cause the fluid over here to move, at first it causes movement of the scalite vestibuli, and then it goes all the way to the tip, and then if it's a high pitched sound, that movement of the fluid with that frequency then gets carried to the base. At the base, then you cause the basilar membrane there to start to move more. As the basilar membrane starts to move more, the inner hair cells starts to bend in that area, allowing us now to hear the higher end. Now, to hear the lower end, we have vibration of the malleus, incus, and stapes that then gets transmitted through the scalae vestibuli up to the helicotrema, and then at this point, right at the very tip, we cause the basilar membrane up here in the scalae tympani to start to shake. As it starts to shake, the hair cells there starts to bend. As it starts to bend, you have an action potential at the tip, allowing us to hear the lower ends. It's kind of cool, right? Now, this kind of explains attenuation reflex that I just mentioned about the tensor tympani and the stapedius muscle. The main job is to prevent damage to the tympanic membrane and the malleus, incus, and stapes that's attached to it. This is a good picture describing why we have the ability normally to hear even the low ends as we become older and older. But the high end of our spectrum we lose really early. Excuse me. And the reason is because of this, right? At the area that is at the tip, the helicotrema, the basilar membrane is much wider. Because it's wider, it's easier to move. Because it's easier to move, it doesn't take much effort, it's very hard to lose that ability to move it. And then it's very hard to lose the ability to hear the lower ends. The basilar membrane is easier to move, making it easier to activate the hair cells, making it less likely that you're going to lose hearing in that range. But at the very base right here, right, the base, well, at the base, the basilar membrane is going to be very thin, right? Not wide. And because it's thinner, it's harder to move that vascular membrane. Because it's harder to move the vascular membrane at the base, because it's so thin, the inner hair cells there can't bend as easily. And you start losing hearing at the higher ends because it's harder to move the vascular membrane, even when we're young. Right, let me show you the animation.